Okay, sir.
Okay.
Yeah. Shall we start?
Yeah, we are in live. Yeah, we are in live. I request the MOC committee to proceed over. Mr. Gokul, please.
Thank you, Ramkan sir. A very good morning to all the dignitaries, guests, and participants with great joy and excitation. On behalf of the organizing committee of Karpagam College of Pharmacy, let me welcome you all to the second day of the International E-Conference on Expanding Frontier of Pharmaceutical Research Towards Global Exigency. As we all know, yesterday we had two of the most exciting scientific sessions conducted by Dr. B.C. Karthik and Sri S. Gurubalaji sir, respectively. Today's session is also going to be no exception. The first scientific session of the day belongs to Mr. Raja Ilangovan, Director, Technical Operations, Lupin, USA. He will be expanding on title Pharmaceutics and Industrial Perspective. Now, I would like to introduce, welcome Ms. R. Gayatri, Member of Organizing Committee and Associate Professor, Department of Pharmaceutics, to introduce our speaker. Over to you, ma'am.
Thank you, Gokul. Good morning, one and all. It's a great privilege and honor to introduce our today's speaker, Mr. Raja Ilangovan, Director Technical Operations. He is here with us to deliver a speech on the title Pharmaceutics and Industrial Perspective. To introduce sir, I'm giving a brief introduction about him. So Raja Ilangovan sir has pursued his undergraduation in Bachelor of Pharmacy from Jesus College of Ooty in the year 1998, and he has pursued his master's in the branch of Industrial Pharmacy from Annamalai University in the year 2000, and he has joined as a operation executive and has started his career in the Cipla Limited in the year 2000 to 2004. He was with Cipla Limited, and later he joined with Aurobindo Pharmaceuticals as an associate manager operations, and he has moved to as a manager technical operations Concord Laboratories, New Jersey, and he worked with that organization up to July 2008. And later, sir has continued his career as a senior process engineer in Ranbaxy Limited up to July 2011. Currently, he is serving as a director technical operations, also known as Novel Laboratories, and now it's currently with Lupin. It is named as Lupin USA. With a small introduction, I will wholeheartedly welcome you, sir, for today's session. Over to you, sir.
Thanks for all the. Am I audible?
Yeah, sir. You're audible, sir. Okay. You can continue.
Yeah. Thanks for all the warm welcome and introduction. It's a real honor and privilege for me getting a platform here to share my industrial experience. As I was a college student, we were blank. We did not know what to how to proceed further, and we know we studied some books and thing. I don't know. We were not having a bridge to connect how to an industry. When I got this thought process, we had lifetime struggle to keep each step forward. But I think your college students are privileged to hear it from somebody who has come through this path to an industrial level and then sharing it back with the industry students. Actually, it is like a no-brainer. You are getting some information which you will have to spend your life to get it. I mean, that is a real-time experience. There's nothing. Nobody can read it in a book because I am the book sitting here, which I went through all these things. Without spending any further time on self-introduction, as they mentioned me, my name is Raja Ilangovan, and I had been in the same industry. I chose industrial pharmacy, and then I chose industry. It was of my specific choice. It was not accident. And then I slowly grew. I am in the position of director heading technical operations in Lupin Somerset here. What does really technical operation does is They do. I mean, we are responsible for all the new product launches here. We are responsible for all the alternate source vendors for any products here. Basically, we are responsible for the lifecycle management of the product. This is what we do here, and we have to provide day-to-day support. All the scale-up processes, any regulatory queries coming, audits, everything has to be supported, and it is purely science-based in the industry. So let me share my screen for the presentation. I was happy to give a topic which is which will connect everybody. That the pharmaceutics books and theory what you read. I wanted to connect it with industry as an industrial perspective. I will not. Don't be afraid of it. That I am not going to teach any physical pharmacy or pharmaceutics or nothing. Professors does it nice, and I'm not going to bore you with any lecture on that. I'm just sharing a real-time industrial experience with all of the students there. I would have loved to give a in-person thing, whenever I'm there, but let time gets through. Usually, when I come to India, I definitely visit Ooty, where I studied colleges, and a lot of my friends are still professors there. So, if time gets, I can definitely stop by your college and then have a one-to-one interaction with the nice administrative team there. And I am familiar with the principal of your college. I know him for past 20-25 years. So let me share my screen right now, and let us go to the presentation what I have with me. Is it good to share it now? Okay, thank you. Okay. Is it my screen is visible, right, Gayatri?
Visible, carry on.
Yeah. So as I mentioned, pharmaceutics, an industrial perspective. So I'm just giving a snapshot of what the industry actually is. Particularly, a country like India, the pharmaceutical sector is actually growing. The Indian drugs are exported to over more than 200 countries in the world. As you are seeing it, it supplies 50% of the global demand of various vaccines, supplies 40% of the generic demands to US market, 25% of all medicines to UK, or 80% of antiretroviral drugs used globally to combat kind of viral things are supplied by Indian pharmaceutical firms. The second largest share of pharmaceutical and biotech workforce in the world, made in India generic accounts for 20% of the global export in terms of volume, making India as the largest producer of generic medicine globally. So you can just call this as India, in other words, that popularly self-proclaimed as pharmacy of the world. India is the pharmacy of the world. So this is just a snapshot of what the Indian the growth sector is. The domestic market is revenue is close to 20.3 billion in 2019 statistics, up 9.8% from the previous 2018. You can see the steady growth of the Indian export market in particular. From 2012, I have compiled it up to 2019. The 2020 fiscal year is going on now, but it has reached close to a $20 billion. Every year it is, the export market of Indian pharma industry is growing. This is a snapshot of the US from United States, which are the who supplies medicines. One is the manufacturing facility for human drugs in US market by country or region. The second one is the manufacturing facilities, by 2020. Here we can see the contribution of India is close to 19 and 10%. One is the second. First is the active pharmaceutical ingredient. The second one is the finished dosage forms. So this 10% market share is gradually increasing every year. The third thing, having said this, this is the opportunity for students thing. When you study pharmacy as a course, and particularly industrial pharmacy as a course, this is the opportunity, because if this much amount of trade is going on, we find a opportunity there. That is why I thought of sharing that. It is not just without a trade, some education systems are working through. That's very important, because we need to know where we will fit in that. Second, this slide is, you can see as you see it, what drives actually it. The center of this, the nucleus of this is the therapeutics, basically the healthcare. Whatever the requirement may be, either it is hypertension or diabetic, or some anti pain medications, or any kind of medications, oncological or any kind of medication, the therapeutics, whatever that decides, everything. Everything else orients that. The new drug discovery, the research and development for formulation development, and once they're all in market, the routine commercial manufacturing of it, then the clinical trials around it, and the regulatory bodies, wherever the global regulatory bodies around it, the marketing and distribution, they all are bracketed under two things. It's like a highway running with two fences. One is the safety of the medication, and second one is the efficacy of the medication. Without this two, it becomes a spurious or adulterated drug. Because remember this, we ourselves consume medicine, and some of our family members are also going to consume it. So this science is nothing like a decorative, or it is not a cosmetic science. This is a life-saving science. That is why it is very important that we are part of a life-saving mission. It's very critical to understand where are we, like we are a linking chain, and we are an important link in the chain. We move to the next one. These are some of the topics, theories in pharmaceutics. You may be studying either in your Martin or your Lackman-Lieberman or anything like that. Any perspective you look at it. You look at, you may come across particle shape, surface area, solubility, specific gravity, surface tension, the crystallinity of that, polymorphism, the moisture sensitivity. I just no need to read everything on this. There are a lot of agents in thing you might have studied: dissolution, diffusion, hydrophobicity, lipophilicity, buffer capacity, complexation and chelating, partition coefficient. You may be surprising at looking all these things, because as such you will be surprised that whether all these topics have an industrial application. If so, are they going to do it during a product development, or doing a routine manufacturing? What are you going to do with this topics which you are studying it? It will be surprising is that the familiarity of this topics is a day-to-day application in pharmaceutical industry. Why I'm touching this is because if you know where it gets applied, you will, while you're studying this as a theory in your school or college books, you will have more connection towards it. I think the very reason for me to present this itself is this is the connection I wanted to make. Anything you need is given in your textbooks. All is expected is for you to make the connections between the theory and what is requirement in that. And I hope I touched that during this short presentation in this. This is just limited number of topics here. Whatever the technical things you studied, I have given that. First thing is that I thought of sharing two classical examples which I have undergone with this. With that examples, you can I can say this, and then you can understand a lot out of this example. The first thing, what the pharmaceutics students or any discipline, either it can be a pharmacology student or either of that. What is needed is that they should understand what is time sensitiveness. Time sensitiveness is in an industrial perspective. What is time sensitiveness? I'm just sharing a classic example with you. So whenever it is a routine commercial routine development product, how the development happens is, you get a brand product, and then you do a characterization of a brand product. That is and then you develop your formulation to match the brand product, and then you do in vivo in vitro correlation studies of that to match your dissolution profiles and other things. Once you are good with that and you feel confident with all the developments, you go for the clinical or bio studies of that. Then once those results are all good, then you do the dossier compilation of that, and then you get the regulatory approval, and then you launch the product. This is the routine thing. But whereas there is the other side of the coin. There is something called a 180 days exclusivity in a market. Why this 180 days exclusivity matters is, if at all, if you are able to hit the first to file, if you are getting approval for a first to file, you get an 180 days exclusivity. You decide your own price. For the 180 days, you don't have there is some limitations for your pricing of the drug. But once a 30 bottle costs in the market of a 180 days exclusivity for $100, after the 180 days exclusivity, 15 companies will launch on the same day, on the 181st day. The price of each bottle which you are selling for $100 will be selling for $2 or $3 now. So this is where real change happens. So in this case, we had an example, which is a live example. Way back in 2012 or something, we were chasing a brand which is going to be generic. It is going to lose its patent. So here what happens, you won't have the brand until last minute. You will not have it for your testing and other things, because you will not have the literature of that, what they used, and unless it becomes the freedom of information is available, you will not able to find out what they have it. So if you have it in the hand, developing is easy, right? Here you don't have it. So here really you need to know what is a molecule, and all your pharmaceutical knowledge comes in now. So depending upon the dose, what kind of excipients you have to go with that, what kind of dissolution profile you need, what is the therapeutics of that, where it is going and working, and what kind of so these are all like a theoretical physicist. You have to work it out on paper. Now we don't have a single formulation to compare against. So what we did was, we were developing a multiple prototype formulation. We don't know which is going to be very closer, because we don't have a reference listed drug, RLD we call it as. So we were developing a multiple prototype formulation, and then these are basically it's a hypothetical release profile. We were assuming that this is going to be the release profile which is required, and this also needs a high understanding of pharmaceutical knowledge. We can't just shoot in air. Then it's a theoretical calculation of in vivo vitro correlations. And when we got the product at 5 o'clock in the evening released, it was procured, and then overnight the dissolution, the in vivo in vitro correlation dissolutions were done, and next day morning we were dosing it for the clinical studies. Out of the 3 or 4 type of prototype formulations, whatever we did it, one was matching very closely. So we did it based on the science-based risk assessment approach, whatever I have mentioned in the slide. So we took that, and then once we dosed it, and touch wood, luckily the bio studies came out good, and then it was matching. We have a formulation now which is bioequivalent to the brand, and then right away coming back and then putting it all together as a regulatory dossier, and then we filed it. We did it in a record of 2 days once the bio studies is done. We have a freedom of information to know who filed next. When we got that information, our filing went at 4 o'clock and 25 minutes, and the next dossier was filed at 4 o'clock and 50 minutes evening. So within a 20 minutes difference, the second product came in. This is what the time sensitiveness means. All the efforts which have gone into this really matters, because for the next 6 months, it's 180 days is basically a 6 months window. The company was enjoying a exclusive period, and that is what basically the bonus or the growth of the company basically is that. So you can understand what you are really competing right now. to this juncture, at this point, when you are working on this, we cannot go and say that let me refer the theoretical book. What is this? What is that? Whether I need this much amount of experiments to understand it. I need that. Evaluations are possible, but not on this juncture. You can do it, but your global competition, you can see that, right? That's what I highlighted here. Need strong understanding and application knowledge of pharmaceutics. This is where you need to connect yourself. Whatever you study, the parameters or the attributes of some systems, you should really understand it, and you have to apply it within no time. And second thing, remember, competition is global and response requires global standards too. We got saved our exclusivity in 20 minutes, meaning the entire globe was working on that. So we are running a race against time, against the globe, and every tools in the world is available for all the other companies which you have it. They too are from a reputed institutes. They do anything you say you have an advantage, the same advantage is there for others too. But science has its own edge. Whomsoever is connected very closely to that and who believes in science, and then they do that, can make a real difference. And that is the example. This is not a not a heard example. This was live example which I was personally involved in that. So this is one second challenge. I hope the first challenge in industry would have thrown light on how time sensitive it is and where your knowledge needs to be squeezed in, and all the applications matters. This is the second example I wanted to share. The first example I chose that was like a developmental example where a product development needs to understanding it. Now the second phase come in, right? This is the routine manufacturing example. So basically, if you see pharmaceutics, there is no classification like that, but overall in industry, they call that as in two phases. One is called as a process development, and second one is like a product development. Once the product is developed, then the process development comes in. So product followed by process. It's hand in hand, one in one, because each influences each. So the technical challenge, I had it, this is my personal example, the first one as well as the second one. The second one is that, the problem I am facing right now is scalability of a formulation. It's failing in dissolution profile requirement. I hope you are familiar with, some of the students may be familiar, and your professors, you can go through that in literature also. There is something called as a factor called F2. This is called as F2 is called as dissolution similarity factor. If you have a formulation with a dissolution of Q point at not less than 85 in 30 minutes, you basically run a profile of that. You take a sample at 5 minutes, 10 minutes, 15 minutes, and 30 minutes. One like 4 or 5 time points in that with the reference product. Either it is with the reference product or with the exhibit batch, which is the dossier batch which we filed it. And basically your batch which you are doing in the scale should match with the same profile. The profile is a belief, because you cannot just say your one Q point is meeting and so your product is equivalent. No, that will not fly. That is the tricky part in any formulation development. You should exactly go in the same way your small scale batch was behaving, and the same bigger scale batch also has to be there. Now, unfortunately, here your number for F2, if it is similar, it will be above 50. If it is not similar, it will be less than 50. Even 49.8 will not fly. It should be above 50. So in this case, the organization has 60 million tablets every year has to supply. And they couldn't scale it up. When I joined there for almost 2 years or 2.5 years, they had been manufactured at the scale of, we call in India as lakhs, but here in this part of the world, they call it as thousands, 650,000 tablets batch. And approximately they are making close to 900,000 batches a year, which is very high numbers. You should not manufacture at that scale. There is no profit in that. increase in every time you issue a batch, and then you manufacture it, dispense it, separately test it, pack it, distribute it. You have to maintain records for all this almost 1,000 batch for that. So this was a challenge to me, and a lot of folks left it like that. I mean, they said this molecule is like that, nobody can treat it, there is a lot of variables, this won't come. So I investigated it from a pharmaceutical perspective, and what I concluded was there are certain variables in granulation so far which were never understood as a variables, right? That lot size of granulation, this we use called as a high shear granulator, and then this lot size matters. Second thing was the conventional, so far the idea prevailing at that point of time, they never challenged certain things while granulating it. My experience was I had done these products with some fluid bed processors in the past. So the uniformity, or the characteristics of the granules, I was comparing it with the top spray fluid bed processors with a high shear granulator. When I found that certain controls were missing here, was like when you do a granulation, your number of ports matters, because you spread it across, you can control your agglomeration. And the nozzles, what you use it, you should have a defined nozzle diameter. Those things were not studied those times, because every nozzle diameter, that is the throughput through which your solutions or anything is going to go for granulation. And the spray pattern into that, the choice of angle with the pressure, you can determine the angle spray. I mean, I'm not elaborating it, I'm just giving certain variables which were studied, and the distance should be maintained between, you cannot insert the gun into the granulation. So these are certain things which I found as a gap, and then I implemented controls over that, and then measured the variables, and then defined the ranges. This is what we say. Once you measure your variable, then you can know how to put a range for that. Because once you put a range, you operate in extremes, and then find out the output of that, you will get the answer for that. So the output was desired granulation consistently achieved, your dissolution. Previously, the F2 was coming around 47, 46 was the best. Believe me, I got it 72, and automatically what happened, the 5.4 million scale became 900 batches every year. Manufacturing got reduced to 120 batches. This itself was a $2 to $3 million savings on manufacturing alone for that site. This savings could potentially write off all the paychecks, bonuses, salaries, power requirements of the plant. That plant has become a successful plant with this one product transformation. You can see what really a formulator or a process engineer with the science, with the pharmaceutical background, with the understanding of the process, and then understanding of the theory. A lot of pharmaceutics goes into this. That's what I have mentioned here, is need a strong understanding of process and application knowledge of the process technology. Here, you have to think little out of box, why this is happening in correlation with your theory. If you read your oral dosage from process product development by Schlegel, he might have given some changes, and even in Martin provides it, and industrial pharmacy, those books provide some dx changes of surface area. What is the when your surface area becomes thinner, how is your dissolution profile, your solubility, your nature of molecule, your everything comes into picture. So that is why I have taken this. I mean, that part is our headache, daily headache. I don't want to trouble you more with that, but this is where your industry will be requiring your knowledge there, and we cannot avoid this, because for 3 years this didn't happen, and this was a thing came to me. One thing I wanted all the my friends to understand this is, in a real time, you may be thinking the time which you are spending in a real day, that is your individual day. 5 minutes you wake up late, or instead of waking up at 6 o'clock, you wake up at 9 o'clock on a weekend, doesn't matter. It's a holiday and it's you alone. But that is not the case in industry. in an industry, 350 or 400 or 500, some industries, 1,000 people are working in an industry, and industry always works in a platform, railway platform basis. You might have seen, train platforms. Their tracks are fixed. The trains come and go, right? For every train, they don't put a new track there. So the timing is very critical, because one train cannot stop somewhere and wait because the other train is not moving out. So to clear that on the track, the value of the time, the amount of every minute there is AHUs are running in the plant, purified water systems are running in the plant, 350, 400 man hours are running there, and any product or process problem is happening is that everything is struck, the following schedule is changing, investigations happens, whatnot. So my say on this point, what I wanted you friends to understand is that value of time should be realized in an industrial perspective. So what is industrial perspective? I have explained you, and then value of time should come from your sound understanding of your thing. Hope you are connecting with the topics, and what is required is there. To put it in a very, very simple language, this is my way of explaining it. Understanding pharmaceutical terminologies in real world applications is, you might have been familiar with this term, y is equal to f of x in an algebraic expression. In a pharmaceutical expression, y is equal to f of x may be a single component system, but whereas in a real pharmaceutical thing, y is equal to f of x1 plus x2 plus x3 up to xn. This is a multi-component system. Simply to say, y is the desired output, what you want. What do you want? You want a first to file product which meets all its therapeutical requirements as well as pharmaceutical requirement. What do you want? You want a product at that scale with its dissolution requirement. So that is your desired output. Y is your desired output. You cannot go and change Y. I'm making a statement again here. You cannot go and change Y. Y is only an observational value. What you can do, you can change only X. X can be 1X or X can be multiple X's. So these X's has interaction with each other. One system interacts with each other. So in the interaction, what can happen? That is the science which you do. That is what you read it. So these are explained in your theory books. Each X, I'm just giving as an example, each X is explained in a mathematical expression. Everything is explained in a mathematical expression. So understanding of the variable input, because each x1, x2, x3 by itself, each are variables, right? So understanding of variable input, each component system, correlation with each other, and estimation of their cause and effect is possible based on sound pharmaceutical science understanding. This, if you are able to understand and appreciate this one part, you can interpret any systems of your what pH does to your system. Now that is one of your x1 or x2, right? Then what is your specific gravity? Anything. So in combination, when pH is 7 or 6.8, what is your specific gravity? So these combination systems, you have a desired output. This is what in a real world you apply it, but on the background, while you're in a college, when you read it, please understand that these are all meaningful variables, and each meaningful variables should be understood in their own spirit. It is not just something a specific gravity. No, please connect to it and then understand what it does to the system and what it does to that. So I'll give a classic example here. Simplest example. I want make things very easily understandable. I don't want a complicated system. Whenever you in the simplest language, you may be familiar. I'm just giving an example. Don't take me wrong. You always come across this term called as a bulk density, right? Bulk density is the amount of material in weight occupying a space. how much material is occupying how much space in a layman language. So it's expressed in gram per cc. How much gram is indicating your weight and cc is your volume. So that's the ratio. So what happens really? If you take 10 gram of material and then throw in a volumetric flask, it occupies now 20 grams, right? So 10 divided by 20, it's 1 by 2, which is 0.5. So it's the unit is mentioning as 0.5 gram per cc. Just reading that number as a 0.5 doesn't help you. What you need to understand from that, you might have been taught in this aspect and you might have interacted with your professors. Pardon me if I am repeating it, but what you need to understand from this 0.5 means in a different perspective is that this gives a little insight into you is one, meaning one person sitting in one chair. That is one. So it is standard. 0.5 means it is 50% empty. 1.5 means it is 1.5% sitting in one person's chair. So now you can connect it to the real world. So density of 1 means normal, a person sitting in its fullest capacity. Half of it is empty. That number 0.5 indicates that half is empty. 1.5 indicates that extra half person is sitting there. Now, if you look at this and then see a powder characteristics, you can make a lot of interpretational difference. Some fluffy material, they will have a 0.35 bulk density. If it is meaning 0.35, what is the meaning to that? The meaning is that is it denser or bulkier? Simple. 0.35 is occupied, 0.65 is free. Whereas some crystalline material, the bulk density will be 1.1, 1.2. It means that it has fully occupied its own space and it is taking some extra space. There is nothing you can create it. This is very, very, very critical in terms of space reduction, because tableting is a space reduction. You convert a bulkier powder into a compact mass, which requires this understanding. A simple term of bulk density, if it is understood in its own spirit, it makes a lot of difference. I'm giving a challenge to you. Solid materials, in terms of atomic physics, you can find it. Solid material like an iron bar, a cube of iron. What is the density of the iron? Try it, find it out. How dense they are packed. It is denser, and it will be interesting. So, simple example. I just took a simple example and then explained to you. Just don't try to read it as just bulk density, void space, tap density. You tap it, gram per cc, 5.5, 0.6. No, understand it is what is 1, what is less than 1. And there are certain languages you can easily interpret it. For example, always you use a term called percentage, right? What is percentage? You may know it is against 100, but even to understand it in a little more closer to your heart, the cent, the per cent, right? It is per person or per cent. That cent you correlate to your cricket players who hit century, right? The century is the cent, nothing but it's 100. The same way when you read number system, there is something called rational numbers, irrational numbers. What is rational numbers, irrational numbers? The term may look complicated. Rational numbers means rationing it. It is possible to ration. You have ration shops in country. You can easily split it. Whatever is splittable is rationable. Irration which cannot split it. Simple. So connect yourself with the terminology, because science terminologies are based on some logic. Connect yourself to that so that you easily understand it, and then in a real world application you can make a difference. I think I took a little extra time here to explain it, but that's what I have mentioned here. Usually expressed this based on sound pharmaceutical science understanding is that these are usually expressed as mathematical expressions with unit dimensions. Anything you find will have dimension, and in a relationship equations. Anything you take it as in a relationship equations. All your Martin book or your Lachman Lieberman, anything, any books you take it, they all are full of equations. Don't get threatened by all those equations. They are simply mathematical expressions of multiple component systems expressing in a multiplication, division, addition, subtraction, exponential, integral, or logarithmic relationship with each other. There is a reason for that, and that is what your professors will explain you. If they explain you, you get it in that, you are a future pharmacist who can make a real difference in the thing. So now, having said to these two examples, I don't want to go much into pharmaceutics thing. I hope these two examples brought to you a sense of industry, what you apply your theoretical knowledge towards the industry requirement. So I'm just moving, maneuvering to the next level. Statistics is no more just kids play. It is almost 50% of the requirements now. I hope the regulators in India for the educational thing, they provide more emphasis for this towards college education, because FDA here, since I'm in this part of the world, they don't believe anything unless it is statistically proved. If you say your process is in state of control or your product is in state of control, there is something called as SPC, sorry, statistical process controls, or statistical state of control. You have to prove that statistically this process, because you put all science into that, right? You developed a formulation, you developed a process, it is combined now, you started manufacturing it. Once you started manufacturing it, now it comes to the real life cycle of a product. There is again the cycle, everything goes there. I don't want to repeat it. There is a scalability, and then after that you make 500 batches a year, 600 batches a year. So there is enough amount of data collected towards that. What you do with that data, that is what the statistics come in. So that is something like that, driving to decision with data. You have plenty of data now. You treat those data in a proper way using a proper statistical tool and come to a conclusion. Very, very important. Very, very important. These things has to be, the skill sets of this has to be developed in the college level, and you will be, believe me, they used to say, the teachers used to say, my kids, you cannot cross 30 minutes in United States without hearing any musical notes. Anywhere you turn on a radio, or even if you put your phone on hold, a music plays. So they will tell that so teach your kids music. So that much is music connected to the same way. In industry, you cannot cross 1 hour without applying any statistical tools. Everywhere it comes. So this is like an unbiased conclusion to prove the output with statistics with an evidence. These are some of the, I'm not going to explain you, don't get afraid of me. I'm not going to take any theoretical class for you, what is that, but certain things which you will be using it, I'm just making familiar. What we use is that this is like a correlation analysis, scatter plot. We use it daily. And expected lower voltages after a flash associated long flash recovery time. Simply, what this is used for, used to illustrate the relationship between two variables by plotting one against the other. That's the principle. That one line, if you know that, you can develop the rest of it. This is correlation. Illustrate the relationship between two variables. Now, as I explained, what is a variable? You can take two variables and then you can illustrate, you can demonstrate what is the relationship between them in the terms of number. As I mentioned, if it is bulkier, what is meaning to that? If it is denser, what is the meaning to that? That number will really speak. So this is the second one. They call this as a contour plot. This will actually show, in a contour plot, the values of two variables are represented. This also shows two variables. In a bird's eye view, you can look at a system. How much is one system influencing the other? Like a map. You can, if you see the geographical map of world, you can see how big Russia is, how big United States is, and how big how small some countries are there. Same way visually, you can make, for example, here in this case, ideal capsule weight is 239 to 252. For a weight range, we have plotted it where their proportions are there, which system is influencing most. where is the most thing. This gives you in a visual way. This goes to the next one. This is also very, very important. Box plots, which we use at daily basis. Here we need to know really in which quartile your data lies. This splits your data into 4 quartiles, and then they call the other ones which comes from the extremes, they call them as outliers, and then they call the lengthy portion as viscous. These are all some familiar names. Don't get get familiar with these names. Here it will say, okay, out of all your data, you have several data. 25% of the data is this is the range. 50% is here. 75% is there. So why these box plots are important is when you set specifications. When you set specifications or action limits, alert limits in an industry, what is action limit and what is alert limit? Somewhere you have to be alerted. If your yield should be not less than 90 or 95, you are routinely getting 98. So your average is 98, but your approved limit is 90. So you cannot have a next batch at 91 and next batch is 98 and next batch is 91. That variability indicates something. That numbers speak something. So something is causing that variation, right? So when you know your average is at 98, you have to split it. How many number of batches? So 75% of the batches, if those data fall at certain point, you choose that as your action limit. If anything goes less than that, that is alerting, you have to take action there. So that this can avoid investigations in industry, in future batches. So this is a tool for that. This is very, very important. You may be, very much aware of it. As I mentioned, bulk density example, understanding a standard deviation is the most important thing, in any pharmaceutics person's life. Without understanding what is standard deviation, the basic one, we cannot step one forward more. The name itself teaches you what is standard, what is the deviation from that. Standard it is like the spot where your data is anchored, where it is standing, where the pole is hoisted. Your pole is hoisted at 90% or 95% or 98%. They call that statistically mean. And then each one, how it varies. From each data, how much it is varying from the mean. If the variation is less, they are all associated close. If it is widened, they are spreaded. Simple understanding. And this is very critical because nowadays there is something called as Six Sigma. It's a certification. I think a lot of institutes in India also provide that. I wish your college also offers those training courses for you. I am a certified Six Sigma person. I am actually training taking up another few projects which will promote me from a Six Sigma green belt to a black belt. It's nothing like a taekwondo or karate or something like that. This is basically understanding the process statistically and then improve process improvement. Some countries have a lean manufacturing. Usually countries like Japan and all, they follow those kind of techniques. US is having mostly Sigma. So process capability, you are certifying your process, or you are estimating your process based on accumulated data with a particular number mark. Here, whenever we read it, we always read with the overall capability and within capability. These are some of the terms whenever you are using it, you'll come to be familiar with it. What is the overall capability? That is the catchword, overall capability. You have 50 batch data. Then within the batch, that is different. But overall, what is your process capability? There is a number to refer. If this number comes above it, it is taken care. Your process is in control. You need not go and justify yourself that it is there. And this is something, a summary report of the calculations what you did it. It's just like it gives finally where it stands. There is something called p-value, t-value. Understanding is, I love to give a lecture on this. I mean, I like that very much. What is p-value, what is t-value. but a lot of experts should be there at your end while they are teaching it. It's, if you know the significance of that, it will be like a watching like a T20 cricket match. It will be very familiar. Nothing thing, unless we don't confuse with multiple things. And these are again control charts. Control charts, as it mentioned that there is an industry, they write something called as annual product report. Every year, suppose if you manufactured 50 batches, 60 batches, 70 batches, 100 batches, you have to write a APR, annual product report. And this APR has to be filed with the regulatory agencies every year. This will summarize the entire performance of the year. So this is a handy tool. You can find seasonal variation. You can find night shift, second shift, first shift variation. If you find some batches continuously, you can find the trend easily. If one technician is getting involved in some some wrong manufacturing setups, so there is a trend there. And if it is a seasonal variation, for example, your winters, India, there is no much extreme weather, but this part of the world is having 4 seasons here. In peak winter, you may get up to minus 18, minus 20, and in summer you will get up to 100, 105. So this range, you will have an effect on the environmental condition. As we know that environmental conditions play a big role in determining the flow characteristics of some of the powders. So a seasonal variation can be caught here, person variations can be caught here, the shift variations can be caught here, and overall the individual value in what range it operates within the variation is allowed. You cannot manufacture anything without a variation, but where is the variation is how much it is running through. So this is a tool we daily use it. Again, this is you may be studying ANOVA on that. Believe me, I don't know even what is ANOVA in my college days. I came to know that there is a topic like that, and then they do something like that. But it is something like I'm not blaming my college days, but if you know the application behind that, fantastic. It's in a simple language. Your analysis of variation used to investigate the relationship between a response variable and one or more predictor variable. For example, you have 5 batch data and one is having outlier. You just want to say this batch is the mean of all these batches is in the same or no. Simple. So if the mean is same, the meanness is the same, so it is not a variable. If the meanness is different, it is a variable. So even you can figure out, again, there is a 1-way, 2-way, 3-way. Don't get complicated. First, let us learn 1, 2, 3, 4. Then we can go to the hundreds, thousands, and all. Here, in a simple way, is that you have a bunch of data and you are figuring it out whether these are similar or dissimilar. Simple. That is what the very topic says. Analysis. Simple analysis. You are analyzing of variance. What is the variance? There is a variance in data. Whether this variance is significant or insignificant. Simple. And you come to a number conclusion. It's like there is nothing like theoretical. You don't need to write 10 pages of summary for that. If this number comes, it is similar. If this number doesn't come, it is dissimilar. Simple. So practice it. There are a lot of tools available in that. We do daily use this, and this is something picking up nowadays. Anybody has a choice of this going to industry, it's very critical, because the formulation development by itself is an expensive process. It is not just mere expensiveness to have it, because you can have 50 variables in an experiment in developing something, but you will not be allowed in industry to do all 50 experiments. And basically, it's a permutation combination, right? It's an exponential, like 3 power 3, 9. And if your variable is 4, it is 3 power 4, or 4 power 4, or you cannot. So this is a handy tool, design of experiment, DOE, which theoretically calculates the which is significant and which is non-significant. So if the signals are not significant, they don't have any interaction in the system. So you can just write up. They are just creating noises to you, and believe me, almost 75% or 80% of your variables can be noises. You can just write off them, and only 20% of the critical influencing one. That's what I have mentioned. In industry, designed experiments are used to systematically investigate the process of a product variable that influence the product quality. After you identify the process conditions and the product components that influence the product quality, you can direct improvement on efforts to enhance. You don't need to do unnecessary things which doesn't have any value to your system and all, unless otherwise you know how to use this tool. Enhance, it can be applied to anybody. It can be applied to a product development, it can be applied to some process improvement. Any projects without this design of experiment, it's not possible. So theory books are available, and you can have some 10 problems worked out with that. How many variables are there, how to calculate their influences, work it out. You will find out that these are all nice projects to have done in your college days. Again, as a student, what I lacked, or at the time which I would have developed myself, was which would have been handy for me in my industrial knowledge, would have been the following thing. I just wrote it. Hope you are all good at that. Don't mistake me that I'm saying that these are not needed for you. But what I wanted to say is that fundamental knowledge of particle size physics is very, very critical. Very, very, very keen and critical on anything. You make a formulation, tablet, capsule, suspension, solution. The particle size physics and sciences, physical chemistry, is the key to your success. You should be master of that. You should know. Here I wanted to emphasize one more thing. Understand the dimensions. That is what I will say. You use terms called ppb, ppm. Understand it. It is parts per million. Million is what? In Indian language, it is 10 lakhs, right? 1 followed by 30 followed by 30. So it is 1 out of that many. The ratio is parts per million, parts per billion. So certain, for example, there is something called cleaning validation. You have to clean your equipment. Just to put in a short way, you cannot allow your existing product which you are manufacturing into your next batch. But at the same time, nobody can claim I have cleaned it 100%. So there is a risk mitigation there. You can calculate it based on theoretical daily dose or LD50, either way, and can consider what is the worst case. They called maximum daily dose, how much it can go. They are all in ppms, parts per million. The max could be 300, but sometimes it is 8 ppm, 10 ppm, 12 ppm, 1 ppm. Same way, I wanted you people to understand what is micron, because most of your active pharmaceutical ingredients are micronized. So they call it as D90. What is D90? Is it something? It is nothing. D90 is 90% of their mean or average is that much. So D90 is 90%. So the most influential is the D90. Rest of it, D10, D50 is 50% and 10% there. So D90 really matters. So micron, 1 by how much? Millimeter, 1000. So just imagine in a micro world how much small it is. So unless otherwise you connect to that physical dimension, it will be very difficult for you to appreciate in a real world what is the influence of that. So get connected to the real numbers: how small, how big, how minute, how what is the diameter of, what is an Armstrong, how small is a nucleus diameter for atomic level there. So get connected with those things. That's what it is. It really matters. It's particle physical sciences in that terms is what it brings to an edge. Then try to get some, I don't know how your industry lab is set up. Have some machines thing, get familiar with the toolings. You go to a mechanic shop, you will find a lot of tools to remove and connect and do that. You should be familiar with that. I became a mechanical engineer when I was doing it, because You won't have a technician on a particular day, and your next day this has to be shipped out. They'll say you run the machine, and that machine produces your product. Only 50% of your formulation and 50% is influenced by the machine. You set up your machine. So if I don't know what is the spanner I should use, what is the screwdriver, what is the thread, what is the size, what is the connection, what is the electric output of it, I'll be a illiterate there. So I need to be familiar with that machine. So it's too late to come and get familiarized with that. Somehow I don't know how that can be designed. Basic understanding of these equipments, not all the equipments, at least the tooling, how to assemble, disassemble some of the things, components, names familiar. What is rear side of a machine? What is front? What is a panel? What is the filter? What they use it? What is the differential pressure in a we use Wooster columns here. How Wooster, you may study those theories there, but what is the differential pressure across the filter? If filter gets choked, what will be the indication? Gun choked, what is the back pressure it shows? These are some certain things, how to assemble it, how to disassemble it, what is the atomization pressure there, how the atomization pressure is influenced by this, and how those connection disconnections works. You can, one way or the other, some colleges can develop some methodology to make the students familiar with these things. It helps you a lot. Then get familiar with the regulatory guidelines, ICH guidelines. ICH guidelines are the most critical. They call the Q series and E series. E series means it's efficacy series. Q series is the quality series. They talk everything: product formulation development, quality systems management. So critical. You may not understand to the full essence of it, but when you are familiar with the guidelines, you will get an industrial opportunity during your course of B.Pharm. You will have industrial exposure for I think 1 month. I don't know the duration, how much is there. We did have it, and you can really find out in the industry how much that is being. You can find, once you know the theory, you can find the place where it is applicable. If you are doing your masters, definitely you can find thing. Including get familiarity with the regulatory guidelines, then drug product types and their key requirements. Overall, you should be knowing that. A diploma in pharmacy will know that, but beyond that, you should be familiar with the types of drug products types and their key requirements are there. Basic pharmaceutical testing knowledges. You should be able to run your dissolution apparatus. I developed my own methods when I was in college. I was doing my project, my dissolution things based on an ultraviolet thing. Identifying the chromophore, what is the lambda max for that, how much it absorbs, what is the Cmax, all those things. Pharmacopoeial requirements. This is very critical. You cannot be a pharmacopoeial illiterate. You have to, what is USP 905 for content uniformity. When you go to content uniformity 905, you should know when the change was done, because your uniformity is the basic requirement of your product. Any product, it should be uniform. So then you know when it was withdrawn. Then there is something called as the ASTM guidance for that, which is the latest one. What is the difference? They use standard deviation and number of samples to correlate that, and then you will have a table there. Once you go to get familiarity with the USP requirement, then you will go to the next step. Just start with one. These are something, it should become like your daily newspaper. USP should be browsed daily, like a daily newspaper. And understanding and interpreting of pharmacopoeial general requirement, documentation and report preparation skills. Here I wanted to emphasize one thing, is that this is one of the key requirement for. I have interviewed kids coming from Long Island University here. Not to mention the university names, what I'm saying is that they are all reputed in Stevens Institute, Rutgers Institute, and someone has completed institutes in Pakistan, someone has done in Malaysia, someone has done in all across the globe. You can find skill sets here. But overall, what I find is that the presentation skills are, applying your writing skills in a scientific language. I have seen maybe less than 10% of the college kids come out and having with that skills. You write it, but the language will be, however, those kind of the languages that it was decided to, however, in the meantime. These are not science. you have to have the skill of writing a report in scientific flavor. Apply each and every line. Alfred Martin book is a classic example. The language, the way it is, going forward, you can read it. And in these are something we can get trained with. There are a lot of online resources available. Just ask your teachers to have some nicely written report. You know the core of it. What you need for that is that you have to read the journals routinely. International journals are there, Indian journals are there. You have to make a habit of reading at least one journal article per day. And in your free time, please discuss with your friends among that how these are there, what are the scientific forum they have referred in that, how the science has been presented well. Believe me, if you have the skill, it will take you through a long way in your life. And again, of course, now I no need to tell that basic computer knowledge, everybody has become a tech guru now, and particularly in Corona time. Nothing else is available for us other than iPhones and other things, computers and that. But I'm not mentioning to watch any, entertainment-based forum here in a softer note. What I'm saying is that get fluency with that. And again, a lot of mini-tab kind of softwares are available, which is easy to use the statistical tools with that. With that said, I want to share this picture. This is one of the greatest picture in human history of so far. This was the scientists meeting, mostly of all theoretical physicists, in Copenhagen way back in 1929. And if you see the names, if you tell in Tamil, like a dialogue, chuma adirudilla and mari, like that. This name, Niels Bohr, Albert Einstein, Max Planck. You are in a day, you step out of your bed and then you go back to your bed. Any book you read, you cannot ignore their contributions. They were the pioneer. Either way, you will find one of their equations. One of that they found it. I live here in US, here close to a town called Princeton. It has the famous Princeton University, and usually Sundays when I go for the close by shopping or something, I always cross the Princeton University thing, and the famous Albert Einstein home is there. I used to show my kids, this is where Einstein lived, and any guest comes to my home, I always take them. I take the privilege of taking them and showing them there, and it is my lifetime thing to see those great names and people who lived it. What I request friends from you is to get familiar with their names, read their biographies, and connect to the scientific world, because there is a history towards it. You cannot be a successful pharmacist unless otherwise you know the origins. Because the theory which you are reading in your books and other thing were invented for some other reason. Max Planck did his Planck constant for some other thing. It was for a black body radiation. From there it came here. Now you use in conveniently for that. Who is Heisenberg? Who is Dept? Paul Derrick. With who is Bohr Einstein thing? Who is Satyendranath Bose? All these names are very, very things. So I wish this picture by itself is one of the greatest picture ever in human history. Get connected with that, and I think I took a little extra time. Sorry for that. I hope I did the best what I could do with the given time. Thanks for the opportunity. Thank you. I'm open if anything is there.
Thank you, Raja sir, for that beautiful presentation. This was more than just a presentation, sir, because the way you engaged the audience by referring to cricketing terms and dialogues was really good. And this was more like a practical explanation to a theoretical questions that the students had. Like you said, why in this world we all studying these terms in pharmaceutics? Almost 80 to 90% of the students might have had that question, but you answered it very beautifully, sir, and thank you for that beautiful session. And now the session is open for questions. I request all the participants to share their queries in the chat box below. And now I request Raja sir to explain, to give some explanations on the query. And I request Mr. Deepak from organizing committee to display the questions of the participants. And there is a first question, sir. In practicals, we used to perform in vitro in simulated gastric pH, whereas in industry, the same is performed in water and buffer in all pH. So what's your perspective on this, sir?
Basically, this is driven by the product. There is something called as a multimedia resolution. If you go to the guidances, there is a separate guidance available for the multimedia. We cannot generalize anything like that. This is basically the site of action where you needed it, and purely the product required. And these are mostly regulated. Industry has that regulation. I mean, you may do it in your college, but they are all regulated. You'll have a fixed pH. You won't do that variables here in industry. That's what my answer.
Yeah, thank you, sir. Deepak, could you display the next question onto the screen, please?
I'll have some water if you don't mind.
Yeah, okay, that's sure. Anything at your convenience.
Second question, Deepak.
Actually, it's 1:30 for me today. So,
yeah. And sorry, sir, we are expecting you in mid.
Yeah, go ahead.
So there's, yes, that's the second question, sir. Okay, this is not a question, sir. This is actually a compliment. It looks good, informative and descriptive explanation. Nicely explained about the connection of a theory and practical and the importance of the time.
Yeah,
so,
Mr. Please confirm with him. Mr. Raj, do you have time? Do you have any other students?
Your voice is mixing. I'm not clear about it. Right?
We will take another 5 minutes.
Go ahead. Tell me.
No. What do you want from my side, sir? One question. How come what are the challenges which are facing from industrial to regulatory approval? What are the challenges of regulatory approval of product?
It's a good question. In the past, people were taking chances. I'm talking purely about US market because that is what for the past 15 years I had been associated directly with. In the past, there were companies taking chances because they wanted to file it and develop it. Any deficiencies during the time, and when they get the question, they say that we have answers for this question, and then they file it. Now there is something 2 or 3 things changed with the thing. There is something called as a GADUFA fees. You pay the Food and Drug Administration for that. So they are bound to answer within certain days. So there is a time-bound response to that. And in turn, what they have done is that they preliminarily screen your application and something called as reject to file. They won't take it for filing. It will be rejected in the first place. So unless otherwise it doesn't meet your minimal requirement, it doesn't warrant that if it is accepted for filing, it goes through. But it has a very good chance. More than 90-95% your application will be approved within. The golden opportunity now is that previously when I was starting working here, it's like a nightmare. When you file it in 2006 or 2007, it will take 3-4 years to get an approval by the time thing. But now it is not that. I think there is a 2 years window within which you should have a pre-approval inspection for that yes/no answers, and then you should go to commercialization. So if you have a sound development and all the supporting data to that, you can have a very short time within which you can go to commercialization. Yes.
Sir, there's a next question, sir. Relating proportionate and realistic combination of theory and notes and practical tips for industrial perspectives from you.
Relating realistic combination of theory notes and practical. I learned it through my experiments, even in my I didn't learn anything through book. That I am very honest with you, and I was not a very bright student. I cannot by heart things. I can understand things. I cannot by heart anything. But some of my friends, including the previous guy who owned that book, they will read that name and by heart that also. Where I learned was basically the two experiments in one of my BPharm project and MPharm project. That is where I learned it. One of my friend and myself, we did it our own on it was a pharmacognosy project in which I didn't do anything much. My friend did it, but I was assisting him in that. That is where that was a analytical related thing. That is most of the analytical related systems I become familiar of that. During my MPharm project. Interesting thing was I was actually this connection which I am speaking to your college is also coming through that one of my friends Saravanan he introduced Professor Gayatri. Then that is how they contacted me to speak to you. We both did the project at the same time. I did it in AstraZeneca way back in '99 in Bangalore, and I developed a formulation which was thrice daily into twice daily with a bilayer tablet technique. It was a 1 year, 7, 8 months project which I did it there, and that is where I actually got into connection with that thing. So I don't know how really I can answer your question at this time. I learned it through experimentation. So your practical design at your college or something like that, do it honestly. Don't try to get answers to that. Do enough mistakes to get the right solution of it. Do mistakes. That is what my answer is. That by doing more mistakes you learn things, and don't feel shy to admit it, saying that this I don't understand it 10 times. Go and speak with your friends, folks, your teachers, and then try to get that understand. That's what I'll say.
Thank you, Raja sir, for your beautiful words, and we hope we've extracted the most out of you, and thank you for the beautiful explanation, sir. Thank you. Thank you, sir. And now I'd like to call upon our principal, Dr. S. Mohan, sir, to share his few words regarding the session. Over to you, sir.
Experience. I'm very sure that all our participants are enjoying with your session. And finally, you end up with stimulating speech. Read about the famous science. That is also very important thing. Origin we have to realize that is a really stimulating speech to all young participants. So, thank you for your valuable time spending our organization. Thank you.
Thank you.
Yes, sir. Thank you, Mohan sir, for your beautiful words. And at this point of time, we are very glad to present an e-memento to our speaker, Mr. Raja Ilangovan sir, as a token of love. I request the organizing committee to share the e-memento on the screen, please.
Thank you. Thank you very much.
Thank you, Raja sir, for your valuable time with us. And now,
just add with all the growing a lot of aspirations. That is that really something is that you will have a great reward. I don't want to tell that if I would not have climbed ladders and then I'm heading in a very hard seat for a technical services in a big organization. certain decisions I take is $1 million decisions. So the rewards are the same way. My salary and other benefits, what I would have been there, and or something like that. I earn 10 years of salary in 1 year here. So I'm just giving a promising words for that. I'll have in a 30 years, 300 years of work for it. They try to get the maximum out of it. So when you put efforts, you will have rewards for that. I just want to add a note. And
thank you, Raja sir, for the words. What's up?
Email ID. If somebody wants to contact me, any help for any industrial job openings or something, that I have a lot of contact friends there. If whatever I could help, I can.
Yeah.
Thank you.
Yeah, thank you, Raja sir. And now I would like to invite Ms. R. Gayatri, the member of Organizing Committee and Associate Professor, Department of Pharmaceutics, to deliver the vote of thanks. Over to you, ma'am.
Thank you, Raja sir. We had a great session today. So the pharmacy as an industrial perspective, I hope all the participants had a great insight into the pharmaceutical perspective inside and in the industrial environment. The speaker nutshelled each and every demand on industry, and he has fulfilled the practical needs as an individual needed to be inculcated from the curriculum. Thanking you, sir, for your tremendous input, as you have provided a good input to the participants, and that I have really would like to mention the time you have spent to us. It is almost 2:30 night, I think so, in your place. So we thank you wholeheartedly from our side. Once again, I thank you, sir, for spending your valuable time. Thank you very much, sir.
Thank you, Gayatri ma'am. And with this, I've come to the conclusion of the session. I kindly request all the dignitaries and the participants to join the next scientific session at 11:15 through the links communicated to you. Thank you.
Okay, bye.
We can sign off.